네, 안녕하세요. 오늘 웨비나에 참석해 주셔서 감사합니다. 저는 젠파마텍의 사업개발 매니저 황신혜라고 합니다. 저희 젠파마텍은 유전자 조작 마우스 모델을 통해 직접적으로 모델을 산업에 공급하고요. 이 모델을 통해 연간 1,000건 이상의 비임상 연구를 지원하는 글로벌 바이오 기업입니다. KUAP 낙가웃 올 프로젝트를 통해서 현재 25,000여 종 이상의 인간화 마우스를 포함한 유전자 조작 마우스를 보유하고 있고요. 미국, 중국에 걸쳐 7개의 자회사를 운영하고 있고, 한국에서는 작년 12월에 성수동에 저희 지사를 오픈을 했습니다. 저희가 서비스하는 비임상 분야에는 온콜로지를 비롯해서 자가면역질환, 대사질환, 그리고 치매나 파킨슨 같은 CNS 질환을 다루고 있습니다.
Okay, thank you for the introduction and thanks for the opportunity. Let us to present our work here. So today I would like to talk about the targeted B-cell therapies, the challenges and the preclinical evaluation considerations for repurposing from oncology to autoimmunities. Well, here is the context I would like to present today. So first, I would like to review the current state of treatment for B-cell malignancy and drug repurposing in autoimmune disease. So we both know that the CAR-T therapy have revolutionized the treatment landscape of hematological malignancy like leukemia, multiple myeloma, and also lymphoma. So the several CAR-T therapies have been approved by FDA. The CAR-T cells that targeting for CD19 and BCMA has achieved a high complete remission rate and high objective response rate in patients. And the TCE, the bispecific antibody, has also emerged as the potential treatment for homological malignancies. The TCE cells the TCE drugs that targeting for CD19, BCMA, and CD20 have achieved the significant clinical benefit in patients. So the successful experience that have been achieved in the B-cell targeting in the treatment of B-cell malignancy lead the scientists to turn their attention to the field of autoimmune disease treatment. So here is a study that published in the Journal of England Medicine that report 50 patients that with severe autoimmune disease. Those patients received one single CAR-T infusion. So after the one single CAR-T treatment, all the patients with severe SLE showing the DORIS remission, and all the patients with the idiopathic inflammatory myositis have the SCR EOLA major clinical response. And all the patients that with systemic sclerosis also had the decrease in the score on the ESTA activity index. And all the patients that received the treatment show the side effect of the patients are minimal with not high grade of the cytokine release syndromes. Usually the main concept for treatment of autoimmune disease is mainly focused on the prevent of the autoreactive B cells from attacking host immunocells by both using immunosuppressant drugs like glucocorticoid and anti-inflammation drugs. But the CAR-T therapy provides the potential method that could deep depletion B cells. So here is in this study, the scientists analyzed the immunocell population after 4 months and 12 months of received CAR-T cell therapies, and they found that the reconstituted B cells showed the naive B cell phenotype. And also the membrane B cells were dramatically reduced. The plasmablasts were not found after B cell reconstitution, and also the SLE-associated active membrane B cells were also not found after B cell reconstituted. So this CAR-T therapy might provide the strategy that to reset abnormal cell population in patients by deep depletion of B cells. And here is another clinical research studies that report the autologic CD19 targeted CAR-T therapy in autoimmune disease. In these studies, the scientists use this UCAR-T to treat the patients that has the myopathy and systemic sclerosis. And the study showing that after receives the infusion of this UCAR-T, the deep B-cell depletion was maintained for almost 2 months. And this UCAR-T can alleviate severe skeletal muscle damage in patients. And also, the UCAR-T treatment can reverse extensive fibrotic damage to clinical organs. So it needed to be noted here. Usually, the extensive fibrosis organ was regarded as to be irreversible. But in these studies, the UCAR-T treatment can reverse the fibrosis organs. So this kind of therapies provides the promising strategy to treat autoimmune disease. And here showing the TCE as application in autoimmune disease. And in this study, the researchers use the TCE drugs that targeting for CD19 and CD3 to treat the arthritis patients. So from this figure here, we found that after the TCE treatment, we could observe the rapid decline of the RA clinical disease score in all patients, and also the patients shows improved severities in Armstrong and PTECT, and also the treatment reduced the autoantibodies in serum. And very importantly, depletion of the activated membrane B cells and the increase of the naive B cells were observed in patients. And here is another study that is also the TCE therapy in autoimmune disease. In these studies, the scientists use the TCE drugs that targeting for CD3 and BCMA to treat 4 autoimmune disease patients. Those patients are suffering from the syndrome, the Sjogren syndrome, the inflammatory myositis, rheumatoid arthritis, and the other autoimmune disease. Those patients are resistant to more than 5 immunosuppressants. But after receive the treatment of TCE, the serum showing the antibody decreased, and also the patient showed the reduced in membrane B cells and plasma B cells. Also, the naive B cells were increased in patients. And also, the TCE drug treatment improved disease activity in all 4 patients. Also, the side effects of the treatment are very minimal when low degree of cytokine release. So here we would like to discuss about the mechanism similarities and difference between oncology and autoimmunities. The autoimmunity and cancer are the 2 sides of the same coin. The regulatory immunocells like Breg, Treg, macrophage, and MDSC are decreased in the autoimmunity disease onset, while an increase of the same immunocell population are associated with the onset and the development progress of tumor patients. And also the molecules that involve in the immunotolerance are decreased in autoimmunity, but overexpressed in cancers. For example, so the drugs targeting to such molecules need to use the opposite strategies. For example, the PD-1 blockers were used to treat cancer treatment, while PD-1 agonists are used for the autoimmune disease treatment. Well, when we compare the treatment strategy between B-cell malignancy and autoimmunities, they share some similarities. Targeting B cells in B-cell malignancy and autoimmunities are the common strategy to achieve disease remission. For the B-cell malignancy treatment, the lineage markers like CD19, CD20, BCMA are used as a target for B-cell malignancy treatment. The mechanism of autoimmunity is complex. Overactivation of B cell contributes to the development of disease. We take lupus as example. The lupus shows the hyperreactive B cells and loss of B cell tolerance. Expansion of plasma B cells will pronounce the autoantibodies, and accumulation of antibodies in kidney cause the tissue damages. So the B cell is also contribute to the lupus development. So the B cell lineage markers like CD19, CD20, BCMA, CD20 are also used for the treatment of lupus and also other autoimmune disease. Then I would like to talk some unique challenges in translating findings in oncology to autoimmune disease. Also, there are some similar between the oncology and autoimmune disease. The preclinical evaluation of drugs in autoimmune disease still poses some unique challenges. The first challenger is from the nature of the indications. There are more than 100 autoimmune diseases. So that means the scientists need to generate different disease models to confirm the studies. Usually, when we do the cancer studies, we can generate different cancer models by using different cancer cell lines and using the primary results as the tumor volume. But when we study the autoimmune disease, we need to generate different models to mimic different autoimmune disease. And each autoimmune disease has the different PD markers and efficacy parameters. For example, if we study the lupus disease, the animal models need to have the autoantibody levels in serum, the protein urine, the changes in immune cell population, and also the renal histopathological change. When we try to study the arthritis, the animal models should show the arthritis clinical score, the pulse thickness, immune cell population, the joint histopathological change. So each disease model needs to have the distinct phenotype and also the PD markers and efficacy parameters. The other challenge for preclinical evaluation of the drugs is from the pathogenesis. The solid understanding of the pathogenesis of the selected models is quite important for our studies. And we all know that there is no one model that is perfect to resemble all the pathogenesis that are observed in patients. Instead, we use different models to mimic the distinct pathogenesis from a specific perspective. So, for example, if we study the lupus disease, if we're working on the c-Gas-STING mechanism, we should choose the models that trigger by activation of c-Gas-STING signaling pathway. If we are interested targeting B cell to treat lupus, we should use the B cell mediated models. And if we are working on PTCL to treat lupus, we should use the BTC mediated models. Also, if we're working on the asthma models, they also have the eosinophil mediated models and neutrophil mediated models. So it's quite important to understand the mechanism of the selected disease mouse model. We need to choose the super models to match with the mechanism of action of the tested articles. Another challenge is from the virus of the therapeutic modalities. There are different therapeutic modalities when we treat of cancer, like the CAR T cell, the CAR NK cell, the ADC, bispecific antibody, the cancer vaccines, cancer treatment. So when we try to transfer this modality into the autoimmune disease, we need to be very careful to choose the suitable models. For example, when we do the CAR cell therapies, usually when we confirm the studies in cancer studies, we just transfer the human CAR cells into the immunodeficient mouse model because of the MHC mismatch. The human CAR cell need to be transferred into the immunodeficient mice. But when we do the studies in the autoimmune disease mouse model, because the disease model is driven by the activation of abnormal immunocells, so the immunodeficient mice is not suitable for this kind of studies. Consequently, the mouse model that used for the CAR cell therapy need to be immunodeficient to receive the CAR T cells, and in the meantime, it need to be capable of showing the overactivation of immunocells. And when we perform the TCE therapy in the autoimmune disease mouse model, because the TCE drugs, in fact, is an effort by activating T cell mediated tumor cell killing. So this model needs to have the functional T cells. In the meantime, it should have the activation of the target immunocells and also the expression of the target proteins. When we evaluate the multiple specific antibodies in autoimmune disease, the models should have the overactivation of multiple immunocells. If these drugs were not cross-reactive with human and murine targets, the mice should also have the human targets by genetic transgenic or by cell infusion. So these mice could have both the immunocells and also the humanized targets. Then in the last part, I would like to give some preclinical evaluation examples of the disease models for the target B-cell therapies. So the first model I would like to introduce here is the SLE patient-derived PBMC NCG mouse model. So in this mouse model, we isolate SLE patient PBMC and transfer the patient PBMC into this immunodeficient mice, the NCG mice. NCG mice is the severe immunodeficient mice. After transfer the lupus patient into PBMC into the immunodeficient mice, we can reconstitute immunocells in these NCG mice. So usually after 6 days of the PBMC implantation, we can measure the anti-double stranding level and also the total IgG levels. The anti-double stranding level is the diagnosis criteria for lupus patients. So in our model, we can display this diagnosis criteria in these models. And the efficacy windows usually last for 3 weeks. During these 3 weeks, we can monitor those 2 parameters in serum, and also we can evaluate the immunocell population like B cell population, plasma cell, membrane B cells. We end this experiment of around 4 weeks of transfer the PBMC and also 3 weeks after dosing. At the end time point, we can do the effects analysis to evaluate immunocell population, and we can test the serum IgG level and also anti-double-stranded DNA level. We can also look at the kidney pathological studies, perform the pathological studies in kidney. In this mouse model, is driven by the lupus patient PBMC. So in this mouse model, it will have the human B cell, plasma cells, membrane B cells. So when we use this model to evaluate our drug efficacy, we can directly to testing for this dysfunction B cells. And as I mentioned, the PD markers and efficacy parameters we can have in this mouse model, including the anti-double-stranded IgG, the total IgG, the B cells, plasma cells, membrane cells population, and also the IgG deposition, immunocell infiltration in kidneys. Since this mouse model is acute mouse model, so there is no proteinuria observed in this mouse model. For this mouse model, we can evaluate CAR T-cell therapy because this mouse is immunodeficient mice. We can transfer human CAR cell and also the other human therapeutic immunocells, therapeutic cells into this mouse model. And since this model has the functional T cells, we can do the TC therapies. And all the immunocells we transfer in these mice are come from humans. So these mice have the multiple therapeutic targets for the humanized antibodies. And we can also use this mouse model for ADC evaluation. Then I would give some efficacy examples to show how we use this mouse model to evaluate drug efficacy. So the first example is a TCE evaluation. The group is like this. The G1 group is NCG mice, is the control group without PBMC transfer. And the G2 group is a mice with the lupus patient PBMC transfer. So this group is our disease model group. And G3, G4 are the treatment group. G3 is treatment when the Blincyto, the TCE testing for CD3 and CD19. And G4 is the treatment with the trispecific antibody. This antibody testing for CD3, CD28, and CD19. So from this data, we can see that after the treatment with those two TCE drugs, the mice showing the completely decreased of the total human IgG. And also the anti-double-stranded DNA level were also decreased in serum after treatment of those two TCE drugs. And we also analyze the immunocell population after treatment of this of those two TCE drugs. The CD20 positive B cells, plasma cells, membrane cells are all depletion by the TCE treatment. And in the endpoint, we also collect kidney to conduct the histological analysis. In this study, we could observe the antibody deposition in kidney as indicated here. So the G2 group is our disease group. And after treatment with those two TCE drugs, those two TCE drugs can reduce the IgG deposition in kidneys. So this lupus-derived PBMC, the lupus patient-derived PBMC NCG mice are quite suitable for the TCE drugs evaluation. Another case we studied in this mouse model is the evaluation of in vivo CAR T and autologous CAR T cells. And here is our group. The last group, this blue column is the we deliver the empty vector, so it's our control group. And this the orange, the yellow one is the Blincyto positive control, and those two groups are the in vivo CAR T and autologous CAR T. So in the first few days, we could observe the TCE drugs could reduce the IgG level immediately. while the antibody levels seems no change after in vivo and autologous CAR T treatments, because in the first few days, there is not enough CAR T cells expansion in the models. But in the late stage, like day 17 and day 20, we could observe the significant decreased levels of IgG level after treatment with in vivo CAR T and also autologous CAR T. Similar results was also found at day 26. The human anti-double stranding levels was also reduced after the in vivo CAR T treatment, and also in the endpoint at day 26, we also observed the significant reduced of anti-double stranding levels after treatment with in vivo CAR T and autologous CAR T. We also evaluate the immune cell population in this model. So after 70 days of dosing, we collect blood to evaluate the immune cell population. From here, we can see that the CD20 positive B cells, the plasma B cells, and also the membrane cells were decreased by the in vivo CAR T and autologous CAR T, as well as the TCA drug treatment. On the day 7, also the in vivo CAR T, the CAR T cell therapy did not depletion the plasma cell, but after 17 days treatment, when we have enough expansion of the CAR T cells, they can decrease the B cell and plasma B cells. And also at the endpoint, we collected spleen to evaluate the deep depletion of B cells in the resident tissues. And we also found that the in vivo CAR T and autologous CAR T can deep depletion the plasma B cells and membrane cells in the tissue like spleen. So this PBMC NCG mouse model that derived from the lupus patient could also be used to evaluate the cell therapies like the in vivo CAR T and the autologous CAR T. Another case I would share here is the evaluation of ADCC-dependent drugs. For these studies, it's also the lupus patient-derived models, but in these studies, we use the NCG humanized IL-15 mice model to conduct these kind of studies. Because human IL-15 can support NK cell survival, since these drugs is ADCs-dependent drugs, it requires the ADCs immune cells population in these models to exert the drug efficacy. And here is the results we get from these studies. We also evaluate the human IgG level and at different time points. The group is indicated here. The first column, the black group, is our model group. We transform lupus patient PBMC into the NCG mice to get this disease mouse model. And from G2 to G6, there are our test articles. We conducted these studies with our clients, so it's like the collaboration work. So we do not exactly know the drug targets. Maybe they're targeting for CD20 or BCMA, or they might be bispecific or trispecific antibodies. So based on this study, it seems like those group is isotype or maybe some single targeted drugs, because this drug shows lower efficacy, while the G2, G4, G5, G6 could reduce the IgG level significantly. And also for the anti-double stranding levels in serum, except the test article 2, the other test article could reduce the human anti-double stranding level in serum. We also perform the immune cell population after 1 week and 3 weeks of administration of drugs. Also, we could find So for the NK cell population, it seems this test one articles maybe infusion with some cytokine like IL-15. So it seems these drugs can help NK cell proliferations, but not the other test articles. And when we look at the plasma B cell depletion, we could observe also accept these test 2 articles. All the other test article could deplete plasma B cells. The B cell, plasma B cells, the CD20 and CD19 B cells seems not as efficiently to be depletions. Maybe those test articles, they are testing for some plasma B cells markers instead of just B cell markers. Okay. Well, here is the endpoint data. We also perform the similar studies in the spleen to try to figure out if this test article could deep replace B cells in the resident tissues. And we have the similar, but we have a little bit different results here. It looked like those 3 test articles could depletion the T cell resident plasma B cells, but those 2 test articles cannot depletion the plasma B cells from spleen. Well, okay. So here is NASH. So if you would like to perform the ADCC dependent drug efficacy that require NK cell population, we can also perform the studies use this kind of mouse model. Well, here is also the lupus mouse model, but this mouse model is the spontaneous lupus mouse model. In this mouse model, we expression BAFF in this mice. BAFF is the autoreactive B cell markers that could driven the mice develop a spontaneous lupus phenotype. So in this mouse model, it will showing the autoantibody levels in serum, the protein urine, the change in immunocell population. Because this mouse model is spontaneous mouse model and chronic disease mouse model, it also showing the lupus nephritis. In this mouse model, we would like to evaluate the humanized antibody that's targeting for CD20. So we're breeding the humanized CD20 mice with this BAFF spontaneous lupus mouse model. So in this mouse model, we can evaluate the drugs that targeting for CD20. This mouse model develops the disease by the autoreactive B cell survivor. So as I mentioned, it will have the autoantibody, autoimmunity, and also have the lupus nephritis. Besides this CD20 BAFF double KI mouse model, we are also generating the BAFF APRIL, BAFF CD3 EDG BCMA, BAFF CD3 EDG CD19 mouse model. This model could also be used for the antibody evaluation and also TCE evaluation. So here is efficacy study we conducted here. The G1 group is just the BABC mouse model, and this green curve is our disease model, overexpression BAFF and also human CD20. And this yellow curve is the treatment group. In this treatment, we use the CD20 antibody, the rituximab, to evaluate its drug efficacy. And from our data here, we can observe that after treatment of this CD20 antibodies, we could observe the gradually decreased level of the anti-double stranding level in mice, and also the IgG level was decreased by these antibodies. We also collected blood and the spleen to evaluate the immunocell depletion. So we can observe the decreased B cells level, plasma cells, and also the MZ blood B cells, the CD20 cells after treatment with these CD20 antibodies. Similar results were also observed in blood. And also this CD20 antibody treatment can reduce the urine protein, like to reduce the UACR and UPCR parameters, and it can also to alleviate the lupus nephritis. As the statistics goes here, it shows the decreased activity index of lupus nephritis lupus. Another model that could also be used for the BCL therapy is the MG patient-derived PBL NCG mouse model. So it's quite similar to that lupus patient mouse model. For this mouse model, we transfer the MG patient PBL into NCG mice and to generate this mouse model. In this mouse model, we can, the PD markers, including the anti-ACH receptor antibody and IgG level. We can also evaluate the BCL population. We can test the grip and also the HE score of the muscle histological changes. So this mouse model can also be used for CAR cell infusion, the TCE therapy, multiple therapeutic drugs, and ADC evaluation. This mouse model, we just have the phenotype present here. We haven't collected the drug efficacy in this mouse model. So in the modern data, we can observe the increased anti-specific ACH receptor antibody level increased in our mouse model, and also increased IgG levels. And also we observed the decreased grip in these mice. And for the histological analysis, we observed increased HE score, like the immunocell infiltration, the fiber degeneration, and necrosis in these muscles. Besides those cases I have presented, there are also the other animal models that could be used for the targeted B-cell therapies, like the BSA-induced membrane nephritis mouse model, the salivary gland protein-induced Sjogren syndrome models, the phoneless MOC-induced EAE model, and also this peptide-induced MG models. So those mouse models are all showing activation of B cells and increased specific antibody levels. So they are both suitable for the B cell targeting therapies. Well, here is the take-home messages. So when we consideration for evaluation the drugs in the autoimmune disease mouse model, we need to characterize the pathogenesis of the disease model. Then we can choose the suitable models to match with our drug MOA. And also we need to know the PD markers and efficacy parameters based on different indications. Usually, we should consider about the levels of serum antibodies, the immune cell population, the clinical score disease index, or the specific organ functions. So it's like if you are working on lupus, you need to pay attention of the kidney. If you are working on the IBD, you should pay attention to the intestine. And also, we need to examine the histopathological change of the target organs. And the other things we need to pay attention is the modality of the test articles. Different modality require different animal models. Sometimes we need to use the immunodeficient mice. Sometimes we should consider about the immune reconstituted mouse model. And sometimes we can just use the genetic engineering to humanize mouse model. And here is our available autoimmune disease models. So just as I mentioned, we should know the mechanism of the pathogenesis and the mechanism of our mouse model. So we labeled the driven immunocells here. So our clients can use these tables to choose the mouse model that match with their drug MOA. Well, here just a brief introduction of our company. I just skipped this part. Well, that's all the that's all the data I would like to share with the audience. Now, I'm happy to take questions.